Cyanine is a non-systematic name of a synthetic dye family belonging to polymethine group. The word cyanin is from the English word Euro-cyan, which conventionally means a shade of blue-green and is derived from the Greek kyanos, which means a somewhat different color, dark blue. Cyanines were and are still used in industry, and more recently in biotechnology. Cyanines have many uses as fluorescent dyes, particularly in biomedical imaging. Depending on the structure, they cover the spectrum from IR to UV. There are a large number reported in the literature. Structure. There are three types of cyanines: streptocyanines or open-chain cyanines, R2N plus equals CH, CH equals CHNNR2, hemicyanines, aryl equals N plus equals CH, CH equals CHNNR2, closed-chain cyanines, aryl equals N plus equals CH, CH equals CHNN equals aryl, where two nitrogens are joined by a polymethine chain. Both nitrogens are each independently part of a heteroaromatic moiety, such as pyrrole, imidazole, thiazole, pyridine, quinoline, indole, benzothiazole, etc. Cyanine dyes. History and use in industry. Cyanines were first synthesized over a century ago. They were originally used, and still are, to increase the sensitivity range of photographic emulsions, that is, to increase the range of wavelengths which will form an image on the film, making the film panchromatic. Cyanines are also used in CDR and DVD media. The ones used are mostly green or light blue in color, and are chemically unstable. This makes unstabilized cyanine discs unsuitable for archival CD and DVD use, as they can fade and become unreadable in a few years. However, recent cyanine discs contain stabilizers that slow down the deterioration significantly. These discs are often rated with an archival life of 75 years or more. The other dyes used in CDRs are phthalocyanine and azu. Conventional cyanines dyes used in biotechnology. Cyanines dyes are usually synthesized from 2, 3, 5 or 7 methane structures with reactive groups on either one or both of the nitrogen side chains so that they can be chemically linked to either nucleic acids or protein molecules. Labeling is done for visualization and quantification purposes. Biological applications include comparative genomic hybridization and gene chips, which are used in transcriptomics, and various studies in proteomics such as RNA localization, molecular interaction studies by fluorescence energy transfer and fluorescent immunoassays. Cyanines dyes are available with different modifications such as methyl, ethyl or butyl substituent, carboxyl, acetyl-methoxy, and sulfo groups. X. Excitation wavelength in nanometers, m. Emission wavelength in nanometers, mw. Molecular weight, qy. Quantum yield. Main C dyes and their uses. Cyanines, C, replace advantageously conventional dyes such as fluorescine, FITC, and hodamines, yielding brighter and more stable fluorescence. C3 and C5 are the most popular, used typically combined for two colors detection. C3 fluoresces greenish-yellow, while C5 is fluorescent in the red region. C3 can be detected by various fluorometers, imagers, and microscopes with standard filters for tetramethylhodamine. Due to inherently high extinction coefficient, this dye is also easily detected by naked ion gels, and in solution. C5 become very popular to replace far-red fluorescent dyes, because of its high extinction coefficient and of its fluorophore emission maximum in red region, where many CCD detectors have maximum sensitivity, and biological objects have low background. The scanners actually use different laser emission wavelengths and filter wavelengths to avoid background contamination. They are thus able to easily distinguish colors from C3 and from C5, and also able to quantify the amount of C3 and C5 labeling in one sample. Other cyanine dyes are useful. C3.5 can replace sulfurhodamine 101. C5.5 is a near-infrared fluorescence-emitting dye. C7 is a near-IR fluor that is invisible to the naked eye. It is used in vivo imaging applications, as well as the C7.5 dye. Sulfo-urocyanine dyes bear classically one or two sulfo groups, rendering the C dye water-soluble, but tri- and quadrisulfonated forms are available for even higher hydrosolubility. Pagylation is another modification that confers hydrophilicity, not only to the dye but also to the labeled conjugate. Nomenclature and structure equals, standard chemical names specify exactly the chemical structure of the molecule. The C3 and C5 nomenclature was first proposed by Ernst, al. in 1989, and is non-standard, since it gives no hint of their chemical structures. In the original paper the number designated the count of the methynes, and the side chains were unspecified. Thus various structures are designated C3 and C5 in the literature. The R groups do not have to be identical. In the dyes as used they are short aliphatic chains, one or both of which ends in a highly reactive moiety such as N-hydroxysuccinimide or maleimide. C dye alternatives equals, many analogues of standard C2/3/3.5/5/5.5/7/7.5 dyes were developed, using diverse modification, Alexa fluor dyes, dialite, fluoprobes dyes, sulfo-C dyes, CETA dyes, Iris dyes from Cyanine Technologies and others can be used interchangeably with C dyes in most biochemical applications, with claimed improvements in solubility, fluorescence, or photostability. C5 ozone susceptibility equals, in 2003, researchers at Informatics and Agilent reported in Analytical Chemistry that microarrays which used C5 were susceptible to intermittent data quality decrease caused by environmental ozone. Exposures to ozone levels above 5 to 10 ppb for 10 seconds were reported to decrease the reproducibility of C5 microarrays. Much higher levels of ozone were required to observe an effect in C3. There are devices that claim to remove ambient ozone levels but they have not been third-party tested. Applications equals. Cyanine dyes are used to label proteins, antibodies, peptides, nucleic acid probes, and any kind of other biomolecules to be used in a variety of fluorescence detection techniques, flow cytometry, microscopy, microplate assays, microarrays. Nucleic acid labeling: in microarray experiments DNA or RNA is labeled with either C3 or C5 that has been synthesized to carry an N-hydroxysuccinimidal ester reactive group. Since NHS esters react readily only with aliphatic N groups, which nucleic acids lack, nucleotides have to be modified with the aminolal groups. This is done through incorporating aminolal-modified nucleotides during synthesis reactions. A good ratio is a label every 60 bases such that the labels are not too close to each other, which would result in quenching effects. Protein labeling: for protein labeling, C3 and C5 dyes sometimes bear typically a succinimidal group to react with amines, or a maleimide group to react with a sulfhydryl group of cysteine residues. C5 is sensitive to the electronic environment it resides in. Changes in the conformation of the protein it is attached to will produce either enhancement or quenching of the emission. The rate of this change can be measured to determine enzyme kinetic parameters. The dyes can be used for similar purposes in FRET experiments. C3 and C5 are used in proteomics experiments so that samples from two sources can be mixed and run together through the separation process. This eliminates variations due to differing experimental conditions that are inevitable if the samples were run separately. These variations make it extremely difficult, if not impossible, to use computers to automate the acquisition of the data after the separation is complete. Using these dyes makes the automation trivial. See also, meroshunine, N-hydroxysuccinimide, maleimide, indocyanine green, J-aggregate, anthocyanin, phycocyanin. References. External links.